Okay, good afternoon. Depending on who you ask, one of the major constraints in getting from your nursery material to the healthy apple trees is plant parasitic nematodes. And once they are present in an orchard and established, they're very difficult to get rid of. So one of the tools in our toolbox to manage plant parasitic nematodes is rootstocks, regardless of the crop. So it is for this reason that we looked at this project, which was the screening of apple rootstocks for resistance against the most prevalent Pratylenchus species affecting apple rootstocks in South Africa. First of all, the culprit here is the lesion nematode or Pratylenchus species. They are migratory endoparasites, so they occur within the roots and they cause severe damage by feeding and migrating through the cortical tissue. So apple trees that are heavily infected with these nematodes have poor growth and the yield declines. And as you've seen earlier, they are also implicated in apple replant disease. Now, in South Africa, a previous survey have shown that there are 3 species that occur on apples. That are more than 80% of the cases, it's Pratylenchus hippeastri. Then we also get lesser amounts of Pratylenchus penetrans and Pratylenchus vulnus. So if we can quickly look at how we did this project. We selected some of the industry standard rootstocks as well as some of the new Geneva rootstocks, and we infected them with nematodes that have been cultured on carrot discs. So during the research, we realized that we get better infection rate if we allow the nematodes to naturally emerge from the carrot discs and infect the roots that way, in place of inoculating them in suspensions into the soil. So our rootstocks were then maintained in a glass house for a 6-month period. And afterwards we extract the nematodes from the roots and count it. We determine root weights, and that enables us to work out the reproduction factor as well as something that we call percentage R. Now the percentage R is the nematodes per gram roots expressed as a percentage when compared to the nematodes per gram roots on a susceptible cultivar. So we look at some of the results. So these are some of the results specifically from Pratylenchus hippeastri in green and Pratylenchus penetrans in the orange there. So if we consider that normally the reproduction factor above 1 is considered the rootstock to be susceptible, we can see in this case that none of these rootstocks that we tested were resistant against these Pratylenchus species, but there were some differences in the response between Pratylenchus hippeastri and that of Pratylenchus penetrans. So we can see that the Pratylenchus penetrans populations that we use are clearly more virulent than the hippeastri. So, seeing that none of the rootstocks proved to be resistant, we did some investigations into the tolerance of these rootstocks, and we did that by comparing the root weights of infected plants versus uninfected plants. And in some cases, in G222, G969, M7, and M793 were the only cases where there were significant statistical differences between the uninfected and infected roots, which indicates that they are less tolerant to the lesion nematodes than the other ones tested. We also did some further evaluations in this regard, and we did that by evaluating the nematodes at 5 existing trial sites where rootstocks were being evaluated for other factors. They were at Paardekloof, Oak Valley, Breevlei, Boeradyn, and Helderwater in Langkloof. And in this case, we collected root samples, extracted the nematodes, and compared the amount of nematodes per gram roots of our subjects to one of the known good hosts at the sites. And we got more or less the same results as in the pot trials. It showed that none of the rootstocks were shown any resistance against the nematodes. But one of the rootstocks, G228, did show much reduced susceptibility than the other ones in these field trials. Unfortunately, during the course of this project, we were unable to obtain tissue culture material of G228 to use in pot trials. So, yeah, just to add, the reason why we only use tissue culture material in this trial is because it was the only way that we could guarantee that we start off with nematode-free materials. Since all of the layer bed materials, 99% of them are already infected with these lesion nematodes. Thank you. That's it.